Hello everyone, and welcome back to this Carbon Academy tutorial on AS level biology. It's been a long time, but I'm back. In this video, we will look at the structure of the cells. So here are the aims of this tutorial. We will identify the structures of prokaryotic and eukaryotic cells, compare and contrast plant, animal, and bacterial cells, describe the functions of the main organelles, and see the features of chloroplast and mitochondria. And finally, we'll identify the gross structure of a virus and its function. So let's look at the structure of the prokaryotic cell. The prefix pro means one and karyon means nucleus. But I decided not to include it as it will confuse you. Clearly, prokaryotic cells don't have a nucleus, but previous scientists thought they did, hence why they named it prokaryote. After discovering that the scientists were wrong, people didn't bother to change the name and stick with it. And there are many examples of this in the scientific community, and I won't bore you with them. Now let's take a look at the distinct features of these cells, specifically the bacteria. A lot of this is going to sound familiar from GCSE, but we just need to add in a few more spices to rig this up to A level. Firstly, as you know, bacterial cells don't have a nucleus. However, they contain circular DNA, meaning DNA starts and ends at the same point. Bacteria contain loops of DNA called plasmids. The DNA of the bacteria is not bound to histone proteins. These proteins will be discussed further in a later video. Bacteria have smaller ribosomes called 70S and have a cell wall made of peptidoglycan. This can also be called murin, but it's your choice what word you like. I personally like murin as it rhymes with urine, but hey, that's me. They have flagella for movement and have pili, which are these spike-like microprocesses that they use for sexual reproduction. The word eukaryotic means well-defined nucleus, and thankfully the name makes sense. Examples of eukaryotic cells are plant, animal, and fungi. Distinct features of eukaryotic cells are a distinct membrane-bound nucleus, membrane-bound organelles, i.e. organelles, like the cells, have a phospholipid membrane. However, do note that ribosomes are the only organelle that have no membrane, hence why they're present in bacteria. Membrane-bound organelles are such as mitochondria. The ribosomes of eukaryotic cells are large, and they're called ATS, and the cells have linear DNA, so they don't start and end at the same point. And the DNA is bound to histone proteins, again which we'll cover in a later video. Unlike bacteria, eukaryotic cells don't have plasmids. A common exam question regards the comparison of animal, plant, and bacterial cells. In this table, I've summarized the key points. Do note that while bacteria don't have a chloroplast, some can carry out photosynthesis via alternative methods. Also note that in animal cells, we have centrioles that make up centrosomes, which are involved in mitosis. In plant cells, the centrosomes are made of a different material. Last but not the least, don't forget the yeast, and excuse my rhyming, as it was intended. And as I mentioned previously, yeast cells are eukaryotic, but they have a cell wall made of chitin, which is different to the cell walls found in bacteria and plant. Now we're going to go through the functions of different organelles, of which many will be familiar to you. Let's start with the nucleus, which contains DNA used in transcription and production of mRNA. Then we have the nucleolus, which contains ribosomal RNA, or rRNA for short, for the synthesis of ribosomes. The rough endoplasmic reticulum, RER for short, contains ribosomes, which is where most protein synthesis occurs. And next we have the Golgi apparatus. And I like to think of this like Jeff Bezos' Amazon. It mainly modifies and packages proteins into Golgi vesicles and is also the site of lysosome synthesis. The Golgi vesicle is a product of the Golgi apparatus, and its function is to transport proteins and lipids to the cell surface via exocytosis. The mitochondria is a site of aerobic respiration and ATP synthesis. The inner membrane of mitochondria invaginate, i.e. fold, to form cristae, and between this is a fluid called matrix. The chloroplast is a site of photosynthesis and is made up of granum, stroma, and lamellae. And this is covered in my A-level photosynthesis video. Link is in the description below. The mitochondria and chloroplasts are peculiar because they have their own DNA. So scientists consider them to be prokaryotic cells, which may have been engulfed by eukaryotic cells like the animal and plant cells. And this is explained in detail by the endosymbiotic theory. You don't need to know this in detail, but having a knowledge of this will give you the edge in exams. Features of these organelles overlap with the bacteria. For example, they have smaller ribosomes, circular DNA that is not bound to histone proteins, and also plasmids. And you can see the similarity between mitochondria, chloroplasts, and bacteria cells. This is the last slide of the organelle functions you need to know. The SER, short for smooth endoplasmic reticulum, is where lipids are synthesized and processed. The lysosomes, which is made in the Golgi apparatus, functions to digest foreign substances or dead organelles using its hydrolytic lysozymes. Then we have ribosomes, which, as you know by now, is a site of protein synthesis. The ribosomes are made up of 2 subunits. This will be covered in further detail in a later video. The vacuole is found in plant cells, which contain cell sap, which is a liquid made of sugars and ions. It helps maintain turgor pressure so that plant cells remain rigid. So now let's look at the cell adaptations and organization. Cells are adapted to carry out functions effectively. For example, if you have many mitochondria, you can produce more ATP, which is useful for a cell that's carrying out active transport, and also useful when the cell is producing large amounts of proteins. Also, having many RER allows for rapid synthesis of proteins and also in large amounts. In terms of cell organization, when same type of cells work together, they form a tissue. Okay? And when different tissues work together, they form an organ. And different organs make up an organ system. Simple, right? Now let's finally look at the structure of a virus. So viruses lack organelles, need to replicate by themselves, and hence they invade host cells. They have an envelope and attachment proteins on their surface to bind to complementary receptor protein of the host cell. Due to its specificity, viruses can only infect specific cells. Example, the HIV virus will only infect the T cell or the macrophage. Viruses that invade bacterial cells are called bacteriophages. So here are the aims of the tutorial. As usual, use them as questions to consolidate your learning. I'm in the process of building a website to consolidate all these videos and add questions. I'll thank you for watching, and I'll see you in the next one. Goodbye.